Hi, everyone, and thank you for tuning into my talk. My name is Maggie Crow, and I'm a postdoc in Jesse Gillis's lab at Cold Spring Harbor. I'm really excited to have the chance to share some thoughts with you about the replicability of cell identities in the Brain Initiative Cell Census Network data, or BICCN data, as I'll refer to it throughout my talk. Since you've hopefully already had a great introduction to the BICCN transcriptomic data from the other presenters in this session, I thought I would focus on the idea of replicability in a bit more detail. So you'll see that my talk is broken into three main components. In the first section, I'll describe the underlying philosophy that motivates my interest in replicability, and I'll provide a brief introduction to our method, MetaNeighbor, that evaluates replicability in single-cell transcriptomics data. Then I'll move on to describe three heuristics that I found to be very useful for this kind of analysis. And finally, I'll show a brief demo of how these lessons can be applied to analyze new data sets. So first, philosophy. If we think back to the basics of experimental design, we know that two of the fundamental components are randomization and repetition. And so these are nicely demonstrated in the classic example, sometimes known as the lady tasting tea. The story goes that a woman once told Ronald Fisher, the famous statistician and of course eugenicist, that she could taste the difference between a cup of tea where milk was poured in first or when tea was poured in first and then milk. And not believing her, Fisher set up an experiment to test this that nicely uses both randomization and repetition. So in each trial, he randomized whether the tea went in first or the milk went in first, and then he recorded whether the woman guessed correctly. He repeated the trials over and over again, and then tried to determine whether the results deviated from the null hypothesis. Funnily enough, in this case, he was proven wrong, as it turned out the woman could tell the difference between the two conditions. She got 8 out of 8 trials correct. Okay, so what does this, T have to do with mouse brain cells? Well, in the setting of cell atlasing, like so many in science, we lack ground truth. That makes it very difficult to design effective experiments, like the T experiment. So the plot on the left is a low-dimensional visualization of thousands of cells profiled in a beautiful cell atlasing study done by members of the Allen Brain Institute in 2018. Each point represents a cell, and the color indicates the cell's annotated type, which was inferred by interpreting the gene expression profiles directly. And so while this was really a mammoth effort, in essence, it really just represents, one trial, like the ones that we saw on the previous slide. So we only have, one guess at how many cell types there are in this region of the mouse brain. And so this is why the table includes these question marks. Since we don't know the cell annotations in advance, it's impossible to determine whether our guess in the trial was correct, though resampling within the test data can help a little bit with this. As a secondary point, it's important to note that integration with existing data can't really help with this because we still don't have ground truth. So integration, like with Harmony or Seurat's CCA method, essentially pretends that we've done a larger trial than we actually have. And one of the risks of integrating is that we'll come up with clusters that don't generalize because we've overfit to the data sets we're analyzing. Okay, so where does that leave us? Well, here is where replicability comes in. Replicability is really the best we can do with imperfect knowledge. So here you can see a table from the previous slide where now each row represents an individual trial or an individual cell atlasing study. We still don't know the true number of cell clusters or if we've made the correct guess, but we can record the number of clusters that we found and then see how those clusters compare to or map to independent data. So in other words, we can evaluate the replicability of the results across multiple data sets. And by comparing many different trials, we can eventually define a consensus. Okay, so I keep saying this word replicability. What do I mean by this specifically in the context of single cell atlas data? And so I'm going to walk through this definition. In general, cell clusters can be thought of as being defined by a collection of genes, some of which correlate highly and some of which do not. For samples which belong to the same cell cluster, the expression levels of these genes are similar, and for samples which belong to different cell clusters, the expression levels of these genes differ. And so this is demonstrated in this plot on the right. Each point here is a gene, and its position along the x and y axis reflects its expression level. And when we compare the expression levels of cells that are from the same cluster, or they have the same sort of morphology and color here, this correlation is very high. but it's much lower when the cells are from two different clusters or are two different types, which is something I'll probably use interchangeably in this talk. Okay, and so finally, the most important part, the relationship between these genes' expression levels for all samples in the cell cluster is found consistently across datasets. So in other words, the relative expression profile should be replicable. And this very basic idea underlies our approach called MetaNeighbor, which I will describe in just a few moments. But before I do that, I'd like to note that this quote is from a paper that is now 13 years old from Tibshirani's group at Stanford. So this really is nothing new or magical for single cell data, but really just a reapplication of an old idea. And I really loved this tweet when I saw it a few weeks ago because it sort of perfectly captured how I felt when I finished writing the first draft of my paper. So on this slide, I'm showing you a schematic representation of how our method works. Our method's called MetaNeighbor. And the whole point is that we want to assess the consistency of expression levels for all cells with the same annotation across data sets. And so we do this using a network-based approach. In this network here, each node represents a single cell. We have cells from three data sets, dataset 1, dataset 2, and dataset 3. The edges between the cells show the similarity in expression profiles, where a thicker line means that two cells are more similar, and thinner lines mean that they're less similar. So we've hidden the annotations for dataset 1, and the question we're asking is how replicable are the expression profiles of the hidden data and the external data? So we score every cell in dataset 1 based on its similarity to the cells in datasets 2 and 3, so essentially summing up the weights of the edges. And then we find out how replicable the profiles are. So once we've done that for every cell in dataset 1, we compare those scores to the known labels to calculate an ROC, so an area under the receiver operating characteristic curve. And if you'd like more details about the method, I will refer you to our paper in Nature Communications. Okay, so that's the end of the philosophy section. To briefly summarize, I've explained how replicability is the best we can do with imperfect knowledge, and shown how cell clusters are defined by relative gene expression levels. I've also introduced MetaNeighbor, our method that can evaluate the consistency of cell cluster expression profiles across datasets. With all of this established, I'm now going to move on to talk about some of the heuristics that we found to be useful for guiding our work. The first heuristic is that feature selection matters. Okay, so what do I mean by this? Let's start by breaking down this plot. The y-axis here is the AUROC, just like I discussed in the previous section. So it tells us about the consistency of expression levels across datasets. In this case, the cluster that we're comparing is GABAergic cells, and we're looking at that across 4 independent datasets. The x-axis shows the results of a simulation where I made this task harder and harder by replacing GABAergic expression with the outgroup glutamatergic cells. And so the key thing to notice is that even at the baseline, where these two cell clusters are as distinct as they possibly can be, there is a huge difference in performance depending on the set of genes that you choose to compare. So if we use what I'm calling an informative gene set for comparison, we'd say that these clusters are perfectly replicable. But if we compare the expression of an uninformative gene set, you'd say that they're barely replicable. And of course, performance decreases as the task becomes harder. All right, so how can we explain this difference in performance across these two gene sets? Well, there's a simple, intuitive explanation for this result, and that's shown here on the left. So we know that some genes are differentially expressed across cell clusters and some aren't. A set of housekeeping genes, like ones associated with the ribosome, are much less likely to be consistently differentially expressed by cluster. So when we measure similarity across these genes, neighbors in the network are not going to be from the same cluster necessarily. On the other hand, if we look at the expression of neurotransmitters, something that's key to this cluster's identity, the neighbors are much more likely to be from the same cluster. So it's very important to choose the right set of genes for comparison. And in our paper, we discuss how we developed a method to select a set of informative genes ab initio, essentially by selecting the intersect of highly variable genes across datasets. And this is what I'll be using as I go through the rest of the talk. The next heuristic is that replicable cell clusters are mutual nearest neighbors. So the data that I'm showing here is from the mouse motor cortex atlas. In this project, we worked with 7 data sets shown here. They're plotted with respect to the number of cells they identified and the average mapped reads per cell. You can see that some methods privilege high cell numbers at the cost of lower read coverage, and vice versa. So there's quite a bit of variability across these data sets. And on the right are some examples of replicable clusters from these data sets. Again, the colors indicate which data set they come from, the size is the number of cells in each cluster, and then the thickness and direction of these edges tell us about the replicability, that ROC score. Okay, so you can see that there's some really clearly replicating clusters here. So SST Chottle is a nice example where all of the clusters are mutual nearest neighbors. You know, if you could see the arrows, which are sort of hidden back there, you'd note that they're all very thick lines, high AUROCs, and that they're bidirectional between each pair of data sets. But on the right is an example that's a bit more ambiguous. So here there are two replicable clusters, CRISP LD2 and VIP HTR1F. You can see that each of those contain data from 6 of the 7 data sets. But then we see there's this cluster in the middle that seems to have similarity to both, even though it's labeled as CRISP LD2. So should, you know, by its name, we'd think it should belong to this cluster. Well, it turns out that this cluster comes from the data set with the lowest read coverage per cell. And so it's likely that this cluster contains, you know, a mix of HTR1F cells and CRISP LD2 cells that weren't properly resolved in this data set. And so in other words, we see that this cluster doesn't have any mutual nearest neighbors, and that helps us to diagnose it as potentially problematic. All right, so finally, the third heuristic is that mutual neighbors with ROCs greater than 0.7 tend to be good candidates. In this plot, the y-axis is the number of replicable clusters, and the x-axis is the ROC, the replicability score. The lines are colored by the cell class that we're looking at, so red for GABAergic, green for glutamatergic, and blue for non-neuronal. And you can see that as we increase the threshold for replicability, we find fewer and fewer replicable clusters. And in our experience, 0.7 seems to be a bit of a sweet spot. Using this threshold in this, motor cortex paper, we reported a total of 70 replicable clusters across the 3 classes in the mouse motor cortex. Okay, so to recap on the heuristics, I've shown you that feature selection matters, that replicable clusters are mutual nearest neighbors, and also that mutual neighbors with ROCs greater than 0.7 are good candidates for further study. Now we're going to get into the final section of the talk where I'll demonstrate how we can apply this philosophy and these heuristics. One of the best ways we can apply these lessons is to take advantage of the replicable clusters from the BICCN and use them as a reference for label transfer. And I'll walk through that in the following few slides. So MetaNeighbor is actually available as an R package through GitHub and Bioconductor, but more recently been working on a Shiny app to make it easy for users to compare their own data to the BICCN reference data. And this is just a screenshot of the web server, and obviously it's in beta right now. We have some placeholder text, so bear with me. So what we'll be doing, as I mentioned before, is using MetaNeighbor to try to map labels from the BICCN to external data. And so here I'll compare data from a paper that was published in Cell in 2017 by Anderben Paul. This data set includes about 500 cells from 6 genetically defined cell populations. And these are sort of representative images of these populations. These cells were selected by hand, either from the primary motor cortex or the primary somatosensory cortex of female and male reporter mice. And they were profiled with a modified version of CellSeq that included UMIs. All right, so now I'm going to play a short video of a live analysis. The Paul dataset has already been uploaded to our server, so if we get started here, we can simply click the Paul to BICCN reference to run the analysis. Let me do that now. In future, users will be able to choose their own input expression data by using the upload feature on the right. The user also has the choice to compare their data at 2 resolutions. Here we're going to pick cluster resolution, higher resolution, and generate results. That'll do the analysis in real time. As we scroll down, we'll see 2 heat maps that summarize the results. These are just 2 different ways of calculating ROCs. Not really important to go over that right now. Each row is a cluster from the Paul data. The columns are cluster and data set combinations from the BICCN. And the color of the heat map is the ROC score. If we look at some clear examples, Paul PV maps up to BICCN PV. We can download these results with the download feature on the right. Great. And then as we scroll down, we can look at some of these results in more detail by looking at this table. So we're going to look at the SOMNOS1 cluster from Paul. We can see its top hits here. In the 7 motor cortex data sets are all SST chottle. So this is a situation where it's sort of arose by any other name. This is the same cluster, but given a different name between the BICCN and the Paul data. Very, very high replicability scores. Okay, so we've now arrived at the conclusions of the talk. I'll reiterate that the philosophy that drives all of this work is the idea that replicability is really the foundation of scientific discovery, whether we're talking about T or cell types. And this is because knowing which results replicate lets us ask why that happens. For example, we can ask, what are the consistent gene differences between clusters? And in fact, that's another key feature of MetaNeighbor, which I didn't really have a chance to discuss, and I'd be happy to take questions on it. We can actually use MetaNeighbor to determine the relative informativeness of various gene sets. In other analyses of brain data, we've shown that genes associated with neuronal communication are very key. Follow-up questions we might ask are, how do these gene differences compare across brain regions or across species, which is something that Nick will discuss in this session. We can also ask technical questions like, how does technology impact this, or computational methods? All of these are topics that are covered in the BICCN preprint, so I will refer you to those for more detail on those questions. Finally, we take this approach because we think that defining replicable profiles is necessary for making progress in molecular neurobiology. We've made all of the data and methods available to ensure the comparability of future data sets to this reference atlas, and I'd be really happy to take any questions that anyone has about the method or about the data. With that, I'd like to acknowledge other members of my lab who have contributed to this work, my PI, Jesse Gillis, as well as postdoc Stefan Fischer, who was one of the primary analysts on the BICCN Mini Atlas project and who has worked to make MetaNeighbor very scalable in R. I'd also like to thank John Lee, who has worked on the Shiny server, as well as previous lab members, Sarah and Manthan, who were involved in the bioconductor package. Thanks to the NIMH for their support, other members of the BICCN. And if you liked this talk, you may be interested in checking out other talks from lab members listed here. Thank you for your attention, and I look forward to answering your questions. Additional references can be found here. Thank you very much.